my channel Solving Questions with Gauri Ma'am. Here we solve questions from IIT JAM examination. So let's have a question for today's video. Which of the following is are involved in the activation of cytotoxic T cells? So the question is from the immunology section, and this is a 2-mark MSQ type of question from paper of biotechnology 2020. Let's see what are the options. Options are MHC1, FCR, T cell receptor and CTLA4. Out of this, which are required for the T cytotoxic cell activation? So let's solve this question today. So let's go through each and every option as we always do and find the answer for this question. So starting with the first option is a MHC1. See, what is this MHC? That stands for major histocompatibility complex. These are a surface protein which are present on antigen presenting cells. What they function for? When antigen presenting cell engulfs the antigen, it carry out the phagocytosis and it presents the part of that antigen on this MHC. Okay? When this part is presented through MHC, it is recognized by the T cells. So the combination of T cells and MHC is fixed. Like if APC is having MHC1, then it is recognized by the CD8 molecule which is present on the T cytotoxic cell. Whereas if APC is presenting antigen on the MHC2, then it is recognized by the T helper cell with the help of CD4 molecule which is present on its surface. So this combination is fixed. Another thing is MHC1 is present on all nucleated cells, whereas MHC2 is present on antigen presenting cells only. Now, in reference to our question, the question is asking about T cell activation. So, here we find the T cell activation in both the cases. So, this is the one of the answer for our question. So, coming to the second option is a FCR. What is this FCR? This is a FC receptor. Okay. See here, when we say the structure of antibody, we find that this is a FC region, okay, and this is a FAB, that is fragment which binds to the fragment antigen binding. We call this as here there is a binding of the antigen take place, and this lower portion is a FC region because of fragment crystallizable, okay. So FCR, what is FCR? This FCR is a FC receptor. Now the receptor for this FC region. The question is asking whether FCR are responsible for the activation of the T cells. So let's see. Here in this diagram, this is a target cell which is having a surface antigen on it. These green colored are surface antigen I have shown, and this is getting attacked by different cells. How these are getting attacked? See, neutrophil, natural killer cell, eosinophil, macrophage, dendritic cells. These are the some cells which are having a FCR. What is FCR? The receptors for the FC region of the antibody. So these are present on these types of cells. Now what happens? First, this antibody recognizes this target cell and it brings this target cell towards any of these cells or all of these cells. Depends. Okay. This FC receptor binds to the FC region of this antibody and the clearance of this target cell take place with the help of all other cells. Okay. For the clearance, these cells may secrete some lytic enzymes, some granzymes may get secreted. Also, tumor necrosis factor can be secreted by these cells for the clearance of this target cell. And in this way, the antibody response is taking place. So if we see here, there is no role of T cells, right? What we have seen: natural killer cell, neutrophil, macrophage, eosinophil. Again, dendritic cells are also having Fc receptors. So there is no role of T cells or T cytotoxic cells even. That's why this is not the answer for our question. Again, I make you remember, when we say this type of immune response, this is called as ADCC, if you remember. Antigen dependent cell mediated cytotoxicity. This is a full form: ADCC. ADCC. This is related to natural killer cell. And again, we study many a time opsonization. Have you heard this term, opsonization? This is related to macrophage. So in both the cases, antibody brings antigen towards the Fc receptor. It gets bound to the Fc receptor, and then these cells triggers the immune response. That's why this is an antibody dependent cell mediated cytotoxicity. Now coming to the third option, that is a T cell receptor. The name itself is indicating that must be having a part in the T cell activation. Okay, so these are present on the surface of the T cell. These binds to the antigen. When antigen is presented, bound with the MHC molecule, right? On the antigen presenting cell, there is a presence of MHC which will present the antigen to the T cell. And T cell receptor is having role to recognize the antigen. These are composed of heterodimer, alpha and beta, or it may be a gamma and delta. Secondly, these are not a soluble. Okay, like B cell receptor, or what we say antibody, that is a soluble protein. That is a soluble glycoprotein that can get detached from the cell and can move in the blood. But this is not the case with T cell receptor. It remains attached with the T cell. Okay. Now coming to the last option, that is D. The option is CTLA-4. This is a cell surface protein. This is an accessory membrane molecule which is present on the T cell, which interacts with the B7 molecule present on the antigen presenting cell. This interaction is required for the signal transduction in the T cell. Okay, so this is not involved in the T cell activation. The full form of CTLA-4 is cytotoxic T lymphocyte associated proteins. So it is having some accessory role in the functioning of the T cell. Yes, the answer for this question becomes MHC1 and T cell receptor. These are the two options which are a correct answer for this MSQ. Now it's time to have a question for my next video. The question is from NAT section. That is a numerical answer type questions are there. This is a 2-mark question from paper of Biotechnology 2018. Let's see what is the question. Both strands of DNA molecule are labeled with radioactive thymidine and are allowed to duplicate in an environment containing non-radioactive thymidine. The number of DNA molecules that will contain radioactive thymidine after 3 duplications is. So question is quite simple. We'll solve this question in my next video. Till then, don't forget to like, share and subscribe my channel and also to hit the bell icon. Thanks for watching.